Good afternoon, everyone. A very warm and hearty welcome to the valedictory function of SVATV, celebrating the success of Genome India Project, and best wishes on the occasion of National Science Day. Every year we celebrate this day to pay tribute to one of the greatest scientific minds India has ever produced, Sir C.V. Raman, for his discovery of the Raman effect, revolutionizing the field of optics and earning India its first Nobel Prize in Physics. This year's Science Day is all the more special as Indian scientists have added yet another jewel in the crown of India's scientific achievement through Genome India Project. Let's begin today's function with singing our national anthem for our beloved country. I request everyone to rise for the same. Thank you. One of the most exciting scientific endeavors of our time, the Genome India Project, is a monumental initiative that aims to map the genetic diversity of our country. It will pave the way for personalized medicine in India, improving healthcare for future generations. However, its success does not rest on scientific advancements alone. It also relies on public awareness and engagement. By understanding the implications of genomics, we can collectively make informed decisions about healthcare, ethics, and policies that shape our society. And that was the aim of SWATV, celebrating the success of Genome India. Before we move ahead, let's take a moment to relive the best moments of this fest. We now present a short video offering a glimpse into the highlights of the past 3 days. Thank you for the video. I now take honor in inviting today's distinguished guest speaker, Dr. Archana Krishnan ma'am, along with honorable member of Vidya Prasarak Mandal, Dr. Mahesh Vedekar sir, our honorable principal ma'am, Professor Dr. Vinda Masramkar ma'am, Dr. Kalpita Mule and Dr. Jayashree Pawar ma'am to come up and raise the bar. I'm sure all of us want I request Dr. Mahesh Vedekar sir to felicitate our today's guest. I now request Dr. Vinda Masramkar ma'am to felicitate Dr. Mahesh sir. I request Dr. Kalpita ma'am to felicitate and welcome our principal ma'am. I'm sure all of us want to know more about how the idea of creating public awareness about Genome India project resulted in SWATV. So I request Dr. Jayashree Pawar ma'am, coordinator of Department of Biotechnology, to take us through a journey of SWATV celebrating success of Genome India project.
Thank you, Dr. Krishnan. A very good afternoon to all present here. It's a privilege to stand here on the occasion of the National Science Day. As is rightly said by Edward Taylor, the science of today is the technology of tomorrow. Likewise, the human genome sequencing perhaps is the most powerful tool ever developed to explore the mysteries of human development and disease. Although the genome sequence of Western populations was determined way back in 2003, the diverse Indian genome started getting decoded in 2020. With advancements in technology, the next generation sequencing platforms, new bioinformatics tools, and relentless efforts by the Indian scientists, a milestone was achieved in the Genome India Project in February 2024 when the sequencing of 10,000 Indian genomes was completed. January 2025 really witnessed the moment when Honorable Prime Minister formally announced this phenomenal success of the project. It was a vision of Dr. Mahesh Vedekar, Honorable Member of Vidya Prasarak Mandal, to design a program and raise awareness about this ambitious project and the landmark victory for students of different disciplines as well as the common people. The seeds of the idea of celebrating the success of Genome India were sown in our minds by him on 14 January 2025. Thank you, sir, for the safety. The idea was thought-provoking. It was a challenge to explain the concept, the methodology, possible applications to the common man. We also felt there cannot be better day than the National Science Day to celebrate this huge success. The activity started with an orientation session for the students, With the help of the students, we try to design posters and models to explain basic concepts like cell, genome, DNA, and sequencing. Stress was also given on the methodology and applications of the project as well as the ethical concerns that are associated. Dr. Thangaraj Sir, the Joint National Coordinator of Genome India Project, and Dr. Prasad from Bhubaneswar, who is also involved in the Genome India Project, scientists working on the Genome India Project, they shared valuable information during the inaugural session. We all have witnessed that. This entire journey has been highly enriching and delightful for all of us. We took this opportunity to build this platform for the unconventional mode of learning for the students, to make it more enjoyable for the participant students, at the same time engaging for those who are learning from the learners. For creating such innovative learning ecosystem, we thought of creating games to attract the naive minds. To satisfy their quest for knowledge, we created interesting games, interesting posters, and working models as well. To reach the masses outside, we thought of creating infotelling event in the form of the street play as a package of information and entertainment together. Through all these learning modes, I hope we have catered to the diverse learning needs. With our students presenting street plays in different areas out of the campus, more than people from different walks of life visiting the exhibit and the positive feedback received, we feel being instrumental in popularizing the Genome India project. We also wish to have many young minds here to be inspired and motivated to pursue research and contribute positively in nation building. Thank you.
Thank you, ma'am. I now request Dr. Mahesh Redekar sir to felicitate a very special guest, Dr. Rakesh Gupta. He is principal. I request Dr. Rakesh sir to come on time. Dr. Gupta is principal consultant, hospital and healthcare, Quality Council of India, Government of India. He is president at CQS, Center for Quality and Standard Foundation, and has developed 16 healthcare long-term formal programs for autonomous colleges. To add, this program includes MSc Clinical Research, Public Health, Hospital and Healthcare Management. He has also developed healthcare modules for Quality Council of India in 2014. Thank you, sir, for raising the occasion. I now request our Honorable Principal Ma'am, Professor Dr. Vinda Manzramkar, to share her words of wisdom with the audience.
Namaskar. Ram Ram. कसे आहात? That's good. Na? So chromosomes are functional. DNA is active. Good? Okay. I welcome dignitaries on dais. Archana Madam, Valedictory Sir, Kalpita Madam, JSD Madam. A warm welcome this afternoon on the occasion of Science Day. We already had begun this way back a week ago. So we were all having these programs. Am I right? We are conducting series of programs every department. You are running, no, you're like my small Doraemons on the back, running and getting the things done, going perfectly. And it was wonderful. You all have shared a lot of new things. That is why today we are having this successful day of science we are celebrating in the college. And yes, we had probably tried to reach the unreached through the Genome India project because it's pure people other than science know what are genes and how they are functional. And when they end up in hospital and then the doctors say, and then they don't write what exactly is going to happen. And then there is so many controversy things happening, and then so that was a good way. I would like, sir, this project we will take to the masses. Since we have our NSS programs, NCC programs, we go to rural areas. Since the things are ready with us, we can propagate this more and more to the unreachables, and we can try to propagate what exactly is happening in our diversified country. Though we are diversified, still we are having the same chromosomes. We are sharing same type of blood with us, which has got hemoglobin and quaternary structure and so on and so forth. And slight differences here and there. That is because of the ecosystem and the dietary habits. So again, welcome on the occasion of the Science Day. Thank you so much.
Thank you, ma'am. It is an honor to now invite Dr. Mahesh Bedikar, sir, Honorable Member of Vidya Prasad Mandal, whose idea materialized into sir.
Good afternoon, friends. I'll not take much of your time because we are eager to listen to Dr. Archana. She is a guest to you, but she is my friend. So thank you so much, Archana, for coming. And biotechnology department needs a big clap, I think so. As we said 1.5 months back, that this idea came in my mind. And all credit goes to that department and madam. It was I had told them 6 months. I said, let's plan something big in 6 months, but they said no, there's a science day, and we will see to it that this project is successful in this 1.5 months. And the most important thing was the students' participation, which we saw. The intention was reaching to students unless and until students participate into such projects. See, this learning was not textbook learning. You had to go on fields. The exhibition was outside, not in the classroom. You had to go out in masses and you had to tell the masses about this project. So such initiatives, this is expected in the new educational policy. As Vidya Prasad Mandal, our all future projects are going to be similar. And I would request you that such type of projects, where students go outside, they get exposure, and that learning is going to be lifelong with them. So thank you so much for this project. This is the beginning. Much more projects are going to come in future with all your participation. One more thing I would like to tell you. I think we are inaugurating a journal right now. So that also is very, very important. I think we must be one of very few colleges who are having such type of journals. And it is an ISSN number journal. So the next thing should be a UGC peer reviewed journal. That should be our target. Maybe next year or in 2 years when we inaugurate this, that should be a news that it is a UGC peer reviewed journal. That should be our vision. So best of luck, and I will not take much of your time. Thank you so much.
Thank you, sir, for your insight and continued support for the development and science and technology and innovation. We move forward, as sir mentioned, about Journal of BNB, JBNB. Science is more than just fact and formula. As we all know, it is a story of exploration, a symphony of ideas, and a force that transforms dreams into reality. To keep this spirit alive in the students and teachers of college, as well as the peers, BPM's B.N. Bhandorkar College has been publishing its very own journal, as sir just mentioned. Since 2013, we have been successfully publishing the JBNB on the inspiring occasion of Science Day. This year, the 13th volume of JBNB awaits to be published. I take the opportunity to invite editor of JBNB, Dr. Kalpita Mohan, to share her thoughts through her speech.
A very good afternoon to one and all present here. It's a great honor and privilege and, of course, a sense of responsibility to stand in front of you
as the editor for the college journal JBNB for the pre-release address about JBNB. It came as actually a new responsibility to function as the editor of the college journal for the year 2024-25. However, with the supportive, encouraging teammates who shouldered this task with me with the same zeal and enthusiasm, the journey till the release of JBNB was truly a memorable one. Let us brief, Let me brief you about the journal. JBNB is a college journal with the ISSN number. It gets published every year on the occasion of the National Science Day. We think that it is our tradition that has to be continued to contribute to the scientific fraternity by responsibly communicating the quality science that performed in the lab to the society at large. JBNB is a multidisciplinary open access journal that gets published annually. For the past several years, the volume of JBNB are made available on the college website for all to benefit. The journal landscapes include full-length research papers, review articles, book reviews, and even the short communications. This year, we received total 6 research papers, 4 review articles, 8 short communications, and after going through a peer review process, total 5 papers, 3 reviews, and 4 short communications are going to get published today in this 13th volume of JBNB. The theme for this year was Science and Technology for Sustainable Tomorrow. My special and sincere thanks to our panel of reviewers. This panel have a few our alumni people who have achieved a remarkable significance in the research area of their interest. And I'm so glad these members accepted our invitation wholeheartedly, supported us, reviewed thoroughly all the requested articles, and gave their critical, positive suggestions to maintain the quality of the journal. I'm indeed grateful to them. Words are not enough to thank Dr. Ashwin Portis, Professor Ames Bhopal, Dr. Gajanan Vade, Sir, Principal, Pillai College, who accepted to function as a member of National Advisory Committee for the journal. Before I request the formal release of this year's JBNB Volume 13, let me also thank all my dear colleagues who are present here in the gallery, the co-editor Dr. Jayashree Pawar, the committee members Mr. Tukaram Naudhare sir, Ms. Kadambari ma'am, Dr. Sandeep sir, Mr. Tejas sir, Ms. Radhika Bhavankar ma'am, Dr. Abhay, Dr. Nilesh sir, and Mr. Rupesh Patil, along with Mr. Nandakishore Sangre sir, for extending their support and performing the dedicated task that made this possible for me for the timely release of JBNB volume for the academic year 2024-25. A big thank you to all of you. May I take this opportunity and request Dr. Mahesh Bedkar, Honorable Principal, Honorable Member BPM, to perform the task of the release of the college JBNB journal. We can just announce and we play the video for the e-release of the journal.
I declare it so.
Thank you, sir. Now UGC care list is no more being carried ahead, so we will have something else to have the benchmark for our journal, but definitely our journey would continue towards the quality. Thank you.
ma'am. Thank you, everyone. Over the past 3 days, we have embarked upon a fascinating journey to the wonders of human genome, knowing about intricate blueprint of life itself. I'm sure the audience, almost all of you might have visited the pandals. If not, I request you to visit it now. Through the informative poster exhibition and enlightening discussions, we have gained a glimpse into the future shaped by cutting-edge genetic research. As we gather here today to mark the closing of this remarkable event, we are in for another intellectual feast with a special lecture from our esteemed guest speaker, Dr. Archana Krishnan. I would like to throw some light, or I am honored to in fact talk about Dr. Archana Krishnan. When I read her CV first, three words came in my mind: a visionary scientist, a true Indian, and an excellent human being. Dr. Archana is a graduate from Ramnaran, India, and after completing her PhD, she is now a co-founder and director of Genomics Limited. The company is involved in developing the medicines for diabetes and cancer. Biogenomics recently launched Inshukvik. This was after 25 years of intense research. Inshukvik is world's first biosimilar version of insulin aspart. We should all give one a big round of applause. In past, he has also co-founded Molecular Diagnostics, now known as Manival Truest. So RT-PCR is a word which everybody knows now after COVID-19, isn't it? Man was pioneer of getting this technology in Indian market. India's first PCR-based diagnostic lab was founded by man back in 2000. Another round of applause. She has designed for 200 infectious viruses and bacteria. The diagnostic tests have been designed by her. Her drive to help India's health system is so high that she refused several offers to study and work abroad or immigrate abroad. So she is a firm believer of innovating and establishing uh, the test or high-end research in India. She is a believer of making India not only bigger, she is working towards it. So we are truly honored to have you, ma'am, in today's occasion. Without further ado, I would like to invite Dr. Archana, ma'am, to enlighten us with her slides. Yes.
Um, First of all, my hearty congratulations to our technology department Whatever I'm hearing from the feedback, I'm actually looking forward to going and seeing the poster. I haven't yet seen it, but after this talk, I'm really looking forward. When I see all of you sitting here, I really am reminded of my college days. So much of energy, ideas, enthusiasm. There is so much that you guys have to achieve ahead in life. And I'm sure the last 3 days have been very, very enlightening because I think that तुम्हाला idea आली असेल ना Human Genome Project मधनं आपण पुढे काय करू शकतो. Do you think you have learned out of the last 3 days? Yeah. What is can someone just kind of summarize? What do you think is like the best take-home message over the last 3 days? What is Manusha? Did you realize that we have a vast array of information available in those four coded words A, T, G, C, right? And there's so much work that can come out of this in the field of biotechnology. And this biotechnology is not just related to diseases or medicine, it can be agricultural, it can be marine, it could be, the applications are just enormous. And what I want to today convey to you is that using the same Human Genome Project and data derived from it, I have built two companies. Okay? And these two companies are purely possible because we have information about the genome. And I'm going to tell you how we do it. Okay? And it's not difficult. I'm sure if any one of you have the drive, have the motivation, I'm sure it is easily achievable. So I will start this talk. Go to the next slide.
Yes.
This is my favorite slide, okay? For people who are little bit new to the field. You can see this mouse here on your. If you see this mouse, on its back is a human ear, okay? And this human ear has been transplanted onto this little child, right? This is the reality of today. There are now chemical-based medicine is changing into genetic medicine. Okay? Genetically engineered organs, like genetically engineered cardiac tissue, kidney tissue is being made, and that can be used for transplantation. There is genetic engineering in the field of diagnostics. You know very well, RT-PCR, all of that is being used very routinely now, correct? So molecular diagnostics, PCR, southern blot, all this you must have studied in your theory, correct? We are looking at first year, second year, third year students. You all have studied all this, correct? So I'm giving you the relevance of what you are studying, right? Gene therapy is a reality, correct? If you know the mutation in the gene, you can also correct that gene by giving it the corrected gene in a particular technology, right? So that's a reality. DNA chips is a reality. So all of this just to show you that what you are studying is very, very relevant. Okay? We know that first of all, within our body there is a cell. The cell has a nucleus. Nucleus has chromosomes. Chromosomes have genes. Genes are made up of DNA, and DNA is made up of bases A, T, G. Correct? We go to the next slide. Okay. Imagine that this genome is like a book. Okay. There are 23 chapters called as chromosomes. Okay. Now in these each chapter contains 7,000 stories called as genes. Now each story is made up of paragraphs called as exons, and they are interrupted with advertisements called as introns. Interesting, no? This I have read it in a book called as Genome. Okay, I found it very interesting. So I always start my talk with students with this analogy. So it's so that you can relate to it. And each paragraph is made of words called as codons, correct? And each codon is made up of letters called as bases. Okay? If I read this, there are 1 billion words in this book, which makes it equivalent to 800 Webster dictionaries. Right? And if I read this genome out to you, one at the rate of 1 word or 1 codon per second for 8 hours a day, it will take me a century. That is the amount of information that is coded into your genome. Now the genome is a very clever book. Okay? It can, first of all, it can, in the right conditions, it can copy itself. So we study DNA replication, correct? And it can also read itself. So it can transcribe, so transcription. All this you must have studied in your molecular biology, correct? Now almost everything in your body, from the hair to the bones to your nails, everything is made up of either of proteins or made by proteins. And what is a protein? It is nothing but a translated gene, right? Now, what I'm going to tell you is, using all this information, how do you, I'm sure you guys, there are TY students who are now at the cusp of brink of making a career choice, correct? And I'm sure you must be riddled by the thought that आगे क्या करना है, correct? So I'm going to give you a light, kind of a snapshot. I'm picturing myself in your shoes 30 years ago, and this is what was in front of me as well. To the next slide, we have now in our country a lot of interesting career options in biotechnology. There's so many institutes, there's so many research labs in terms of private companies who are doing some real state-of-art work. And you, as scientists, I'm sure if it has, the curiosity has peaked in the last 3 days, there's some really, really interesting research work that awaits you. Okay? So go to the next slide. I personally decided that I will go the entrepreneurial route. Okay? We go to the next slide. I chose medical biotechnology as my career option. And so when you say medicine, that means it could be either it could be therapeutics, that is making medicines for curing a disease, or it could be in diagnostics. So I'm going to talk about the two fields that I chose. In the field of therapeutics, I, with the help of my two colleagues, established Biogenomics. Biogenomics is a company that makes recombinant human insulin and analogs. It's a treatment for diabetes. And also, in 2000, we founded Molecular Diagnostics, which was India's first PCR lab. At that point of time, even doctors were not aware that this can be used. There is a technology called PCR, which can be used for diagnosis of infectious diseases, of cancers, of genetically inherited diseases. So I established a company called as Molecular Diagnostics, which later on went to become iGenetics and was further acquired and is now under the brand name of Molecular Diagnostics. Go to the next slide. I was schooled in the Rotary campus in Inauguration and graduated from India College, and then did my post graduation from MS Kolkata, where I won the gold medal. After that, did my PhD at IIT Bombay, but that's when I decided that I will not go to India. I will stay here. And that was a time when medicine was actually moving towards personalized medicine. And stem cell therapies was new. And some of the greatest developments were coming from biotechnology. But having said that, in our textbooks, we have in the 1980s, there are international MNCs who had made insulin and were selling very, very expensive insulin to Indian patients. So that is when I thought, okay, why can't we make effort and make indigenous insulin which is affordable to an Indian patient? Okay? Because cloning and gene expression was our expertise. So why not do that? Uh, but in order to do a huge project like this, there are several challenges. And little did I know that I was as enthusiastic as all of you. Little did I know that we're going to have to face a whole lot of challenges. Go to the next slide. Uh, Start with funds. Who's going to fund my research? Correct? There are so many high investments required for developing a technology like that. I need good talent to do it. I need to know what kind of products I'm going to focus on. And is there a market? Are these products going to sell at the end of the day? And then what is the regulatory requirements? Like, it is ultimately a human injectable. It has to be safe. So regulatory requirements were intense. And how will I reach my product to the final diabetic patient? So these are few of the questions that I started with. And go to the next slide. Uh, we started with this aim in mind, a company called Genomics. Genomics was founded in 2002. So we have today a total strength of about 200 plus, fully funded by international investors, and a state-of-art research facilities where we do everything in-house. Now, in the we have basically specialized as a diabetes one-stop shop portfolio, where we make biosimilars. I will explain to you what biosimilars are and how Human Genome Project will link that with recombinant therapeutics. Go to the next slide. Quick, this is basically the three of us, Dr. Sonal, Abhishek, and myself. Three of us, we started the biogenomics as a concept. If you go to the next slide. Yeah. Now, recombinant very early on, people used to take pig insulin. Human insulin in giving it as a treatment was practically not possible. Human insulin is made by pancreas. How will you get human insulin? Unless you have a pancreas, human pancreas, and you isolate insulin. So people used to take animal insulin, pig insulin or cow insulin, alright? But ultimately that insulin is animal origin insulin. So your body, if you inject your body with animal insulins, after a period of time, your body is going to destroy it, saying it is a foreign insulin. So what is the next best alternative? This human insulin. Correct? Now, where do you get human insulin? Because of recombinant DNA technology, because of Human Genome Project, the insulin gene sequence was very, very well known. Now, if I know the sequence of the insulin gene, then what I can do is I can synthesize this gene in the lab. Okay? Once I synthesize this gene in the lab, it is literally taking this gene and sticking it into a bacteria. It's literally like a tailor's job. Cut out, enzymes, use enzymes, cut it, and then you stick this insulin gene and paste it. Okay? Now you have a bacteria making human proteins. So can you imagine the wide array of human proteins that can be synthesized? You don't need a human pancreas to isolate insulin. You just need insulin gene sequence or any gene sequence. And how is that possible? Because of course, Human Genome Project has been able to sequence the entire human genome. Correct? And that is how the entire evolution of recombinant DNA therapeutics has occurred after the Human Genome Project was successful. Now, in the 80s, we had a few Novo Nordisk, Eli Lilly. These are all the big giants in the diabetes industry. They had come up with insulin, and in India there was nobody producing. we're all patients are taking pig insulin and cow insulin. But all these companies, once they launch their product, after 20 years, their product patents expire and they become generics. You know, biologics, when their patent expires, they become biogenerics, but they are also called as biosimilars. And that's a huge opportunity for us to have to launch these kind of medicines in India so that they become affordable for our Indian masses. So this is a list of several molecules that have now become biosimilar. We go to the next slide. But to develop a therapeutic is a long process, right? Drug discovery, people say, takes lots of dollars, lots of time, and doing it does. But it's a game of patience. So once you clone your gene in the E. coli, in a shape, you see it's growing well, and you run a SDS page, you see it's expressing, correct? Then what you do is, you want to now scale it up. So you grow it on a larger scale, on a ferment. You studied fermentation, industrial biotechnology, in a curriculum. Fermentation technology, you grow the clones and you make them express your insulin. From there, you purify it. So you develop the entire process from there. And once the insulin is made, you have to formulate it. Correct? You have to put it into the vials or cartridges. Correct? So you formulate it. Once this insulin formulation is ready, you have to take it through a series of regulatory processes called as animal studies. First, show that my process, my insulin that I have developed, is safe in animals. No rabbits died, no mice died, no rats died. Right? So first establish safety in animals. Then what you do is a phase 1 clinical trial. So how many of you have a background on clinical studies? Right? So if you don't, you will study it eventually. It's a very important part of drug discovery. So you establish in diseased person that this insulin is actually behaving like it is reducing the blood sugar. So all these studies are first done on patients. Once this is done, the data is submitted to the regulatory authorities. That is when you get your regulatory approval. Go to the next slide. These are the range of insulins that we have developed. So there are several types of insulins. It's not just human insulin now. It is a fast-acting insulin. It's a slow-acting insulin. So there are insulins which are given at night time. There are insulins which are given post a meal, right? So there's a range of insulin that we managed to develop after that. Go to the next slide and the next. So all in all, the team, we started off with an idea of making recombinant products, and we really specialized and we made a name for ourselves mainly because of 5 unique points that we had. Number one is during the development of these products, we studied the process so well using concepts of quality by design. These are very important concepts in the pharma industry. Quality by design is when I do an experiment in a shape flask, in a lab, and when I scale it up on a fermenter, I should get exactly the same results. They have to be reproducible in terms of yield and in terms of quality. Quality by design becomes a very important parameter. So we use robust principles of quality by design to develop these products. And of course, none of our products we have borrowed technology or we have bought technology. It's all developed by us ourselves. It's 100% make in India. I'm very proud to say that. We have a very smart team of scientists. They're very highly motivated, just like you kids. Once we made our insulin, we had the innovator insulin and ours was a biosimilar insulin. We had to show that it is exactly same. So it's like a fingerprint. Because protein is a huge molecule. It folds. It has primary structures, secondary structures, tertiary structures. All that we have to show an exact overlap to show that this is a biosimilar molecule. So being like a fingerprint, you need to characterize it. And how do you characterize it? You must be learning all those techniques in your theory classes, right? So things like amino acid sequencing, N-terminal peptide mapping, correct? All these circular dichroism. I don't know, have you studied all this in biophysics? You have, right? You heard of it, right? All that you are studying is very, very relevant. I'm trying to tell you this. Okay, so please focus when your teachers are teaching you the very important concepts. We study the higher order structures and also study purity, do purity analysis. What kind of purity? If there is any impurity coming from my process, characterize that impurity, study that impurity from all sides. Now, before you get inside a human being as an injection, I also need to show in vitro in the lab that this insulin is lowering the blood sugar, can lower the blood sugar. So you have to do biological assays. You have to show in the lab by growing cell lines of say liver cell line or adipose cell line, you have to show that it's behaving exactly like the human. So since it was so well characterized, if you see, this is the pharmacokinetics and the pharmacodynamics curve. So exactly overlapping PK/PD curves we got. And of course, to build all our facilities.
I'm so sorry.
Yeah. So you can go to the next slide. And as we also innovated, we kept on patenting our work because we don't want anyone to tell us that you can't use this technology because this is something that we have. And at the same time, we raised money and built facilities. We go to the next slide. This is our plant in Patanjali where we have, this is a clinical, a pilot plant which made clinical material. Go to the next slide. You see, anything that you want to do good quality biotech research, we have it. So we have cell banking, we have fermenters, we have LCMS, we have capillary electrophoresis, we have surface plasmon resonance. You know what surface plasmon resonance is? So when insulin binds to receptor, the binding kinetics, the PK, PD, everything is studied using surface plasmon. Okay? Go to the next slide. This is a kind of a snapshot of our Patanjali facility. So this is the fermenters, the centrifuges, homogenizers, lots of other small equipment that we do. Go to the next slide. This is column chromatograph. All of you studied column chromatography and exchange, site exclusion, all of that is used for the field. You go to the next slide. Go to the next slide, please.
Ma'am, earlier one.
No, next. Next.
Go next.
So this is kind of a snapshot to show you how fingerprinting was done. For instance, the amino acid sequence of the innovator and ours exactly matching, or the mass was found matching exactly. The amino acid sequence was found exactly matching. And so on and so forth. So it just keep going. So this is like, I wanted to just give you an idea that all these techniques that you are studying are all used here. So knowledge that you are gaining today in college is going to be very, very useful if you choose a career in this field. Okay? Go to the next slide again. HPLC based analysis and go to the next. And now you know about how insulin actually acts inside the body, correct? Insulin binds to the insulin receptor of the cell. This receptor then triggers signal transduction pathway, which recruits glucose transport receptors to the surface, which transports the glucose inside. Alright? Now, you have to show that my insulin is able to do this in vitro. So how will you do that? You will culture um, liver cells, a hepatic cell, right? If you go to the next slide, and then you will add the media containing glucose, primed with your insulin, and there will be immediately an uptake of insulin from the media. So very simple assays, very application based, but developed so that we can demonstrate how the molecule is going to do its mode of action. If you go to the next slide. We also know that insulin also makes adipogenesis. So you see when the cells which are not treated with insulin and the cells which are treated with insulin have made lipid molecules because of which it is now taking on the stain, oil red stain. The lipid molecules get stained by this chemical. So this is some of the examples just to give you a kind of demonstration. Go to the next. Go ahead. There are further more concepts which are a little bit more terse, and maybe I'll skip all this and go straight to now the diagnostics. So I talked about all the therapeutics part, but PCR आपको सबको पता है ना, RT-PCR, correct? So in 2000 we launched about 200 diagnostic tests. All these tests were mainly based upon detection of infectious diseases, genetically inherited disorders, and various cancers. And I'll talk to you about a couple of very high-end tests which we developed. Go to the next slide. Yeah. So what is molecular diagnostics? They are diagnosis of diseases which are difficult to detect, in a timely manner, and which also can characterize organisms like genus and species well. So some of the very interesting work that we did is the viral diagnosis, which is in HIV, HPV, and HCV. Then tuberculosis, a big issue in India, and how we addressed tuberculosis using PCR-based technologies. In fungal diagnosis and prenatal genetic. Okay, go to the next slide. Now you know HIV, especially in 2000, was a huge problem. HIV, the antiretrovirals were not so well developed at that point. But because of the Inauguration project, we know the HIV gene sequences. We know that subtype A is more aggressive than subtype B and subtype C. Subtype A infection may you need to get treated with a very aggressive antiretroviral treatment compared to B or C. Right? So this knowledge is very important for the physician to start administering the treatment. So this was some work that we did. If you go to the next slide. We actually not just detected it, we also sequenced this information and gave the physician the subtype that he's looking at. And several samples were actually analyzed at our place, and we have a fair amount of mapping of which region. So you can do a lot of epidemiological studies as well. So which region, which subtype is more prevalent than the other. Go to the next slide. Not just that, you can also see the antiretrovirals are targeting either a DNA polymerase or an RNA polymerase. You can also see if there is a drug resistance that is developing amongst these subjects. So, some very advanced diagnosis of HIV we were able to do using the sequencing, DNA sequencing. Go to the next slide. The fungal detection. You know, now today fungal detection is only done using microbiology, correct? And to grow a fungus in the pathology lab takes about 40 to 50 days. And then most of the times the fungus doesn't end up growing and you could get a diagnosis. So here is a PCR within 24 hours, even if there is one fungal spore present, it can very accurately diagnose, and not just diagnose, it can say this is Candida albicans or whatever, it can give you the genus. So this is another very interesting test that we developed. Tuberculosis. Tuberculosis in India, I was in molecular diagnostics when American scientist had come there. And he said, Madam, he was a big scientist in tuberculosis. He had done a lot of work. And he told me, I have never seen mycobacterium tuberculosis. I was like, you have such a big name and you've never seen it. Because in abroad, these bacteria you don't get to see. Whereas in India, you just sit in the local train, go to PT and come back and test positive. But our exposure is so high. We are always sitting in a 37 degrees incubator here. So it's always growing. So making these tests relevant to our country becomes very important. So just a DNA-based PCR is what usually these kits offer. And all will test positive if we use those kits, correct? So if you go to the next slide, we introduced something called as a viability PCR. So a viable bacteria will have the DNA, will have rRNA, and will have mRNA. A dead bacteria will have the DNA and the rRNA. And dormant bacteria will have the dormant will have the DNA and the rRNA, whereas the dead one will have only the DNA. So if you want to detect viable, which is a relevant tuberculosis bacteria, take the mRNA. We develop the mRNA. Correct? So this is some interesting examples that we had worked on. Go to the next slide. Prenatal diagnosis, and I'm sure Mahesh will appreciate this a lot. But uh, it's such a big issue of prenatal diagnosis. Just the blood test that we do makes the patients very anxious whether the baby really has any genetic disorder, right? But we have known from literature that after 15 weeks of pregnancy, there are about 1,000 to 2,000 fetal cells circulating in the mother's blood. What if I'm able to capture those fetal cells and screen for some important genetic disorder? So these are some very interesting technologies we have established using magnetic beads, antibodies on magnetic beads. We have captured the fetal cells and we have tested for all these genetic disorders. Again here, why is this possible? Because we know the mutations because of the human genome project. Okay, so I want to tell you that all these fantastic work that has happened in the field of DNA sequencing of the human genome has a wide range of applications. What I spoke about was just probably nothing. Very relevant question, PCOD and PCOS. Very rampantly seen because of our lifestyle and our eating habits and everything. So, PCOD and PCOS, although the genetic mutations don't occur in this, right? But there are some kind of epigenetic changes, right? Which can influence and can cause these kind of disorders. Now, this is one step a little above diagnosis using epigenetic mutations like methylations. These kind of changes that take place on the DNA. Yes, it's a very relevant diagnosis. I don't think anyone's working on it.
Yeah, what is the difference between this is humulin, which is developed by LIT, and insulin developed by Biogenomics?
So human insulin, they have the human insulin is what is it? The insulin found in our body. So this is the pancreas. Okay. Now this human insulin helps in blood sugar control. However, it can be made slightly better because if if you eat at time zero, the human insulin will start its secretion, start its action after 20 minutes. Whereas if you have smarter versions of insulin, like the insulin that we have launched, that can act within the first 5 minutes. Okay, so fast acting insulin. Then there are insulins which are taken in the night. So in the night, there is no insulin front. You're not eating, but your body is doing basic metabolic functions like respiration and all the organs are functioning. So that also will require blood sugar, correct? So there are nighttime insulins as well, which are slow releasing. Slowly the glucose is released into the bloodstream. So all these variations of fast acting, slow acting, ultra fast acting are all actually mutations of the human insulin gene. This human insulin gene is tweaked in a few places where some amino acid is replaced or something else is happening. That's how it is. That is the difference between the human by LIV and
Because of the allele, there are some slow metabolizers of the drugs, some fast metabolizers of the drugs. So is that point also considered, or are there any tests available for detecting this so that we can modify the dosage as a pharmacology?
Ma'am's question was that because we all have various different types of genes and alleles, some of us may be metabolizing a drug faster than the others, correct? And is there any study, or do we have to do any study like that? Unfortunately, we don't have to do a study like that. It's a very relevant question. But what we have to do is take a set of statistically significant populations. So I would take 350 odd patients and do the same study and show in statistically significant manner that the drug is able to metabolize. That is what is available. However, for specific drugs, the pharmacogenetics is a highly specialized field and there's lots that get explored.
Excuse me, can I ask you a question regarding the role of this science in diagnosing cancers?
Cancers, yes. The diagnosis of cancers is very, very interesting. Now, some cancers are caused by viruses. Like ovarian cancer, there is human papillomavirus. Some cancers are caused by genetic mutations. Some cancers are inherited. For each cancer, it is a different type of diagnosis. For breast cancer, there are some clear genes called as the BRCA genes, which the breast cancer genes. So there are BRCA1 mutations, BRCA2 mutations. Some of them respond to the receptors called as HER2 receptors. So those we need to look for those mutations so that we can decide the treatment whether there is a drug called as Herceptin, whether it can be used or not. So breast cancer is handled in this particular. Ovarian cancer, you look for human papilloma virus integration into the cancerous genome, into the biopsy sample. So if you can find out, if there are especially there are 64 serotypes of human papilloma virus. Cervical cancer, sorry, yeah, cervical cancer. 64 types. And those serotypes, there are 4 or 5 which are particularly very, very oncogenic. So we look for those. So that is, each cancer has a different way of, it has a different way of diagnosis.
I wanted to say that few of the cancers are new genetically.
Yes.
So can there be some genetic engineering to help in preventing necessity to reduce ovarian family, Chinese CFS, or next ovarian cancer. Correct. That needs to be implemented.
Yeah.
Before it happens, it should happen.
So before it happens, today for breast cancer, we could just go into a cancer.
That's all.
So, but I don't know if you've been reading the updates today too. But there's some very relevant research happening in cancer diagnosis today. In that, the mRNA vaccine that we took for COVID can now design an mRNA-based vaccine for cancer. So your body will become immune to that cancer. If it develops, it will be destroyed. Right?
So Russia has been training this.
Yes, it's going to be a groundbreaking. I'm sure it's going to be a Nobel Prize winning diagnosis. If cancer cure comes using vaccines, that will be absolutely groundbreaking.
One more question, a last question. Regarding this Guillain-Barré syndrome, DB syndrome, which has recently erupted, started with Pune. The reports are severe disease in 50 years, I saw only 3 cases, and now suddenly there are hundreds and hundreds of cases. But it has been on the run, South America. And the theories we will talk that it was due to virus, now it said it is autoimmune. See, your whole system attacks it. So how can the science help in this? At least the early treatment of growing of it. So no residual damage is happening.
This is an autoimmune disorder, unfortunately, and the only way of preventing further damage is to prolong the illness. So that is the only way of using steroids. That's the only way of treating it as of now. What triggers it is a question that is more important and relevant. And why is it getting triggered in a few patients and not the other?
And about 5-6 bacteria.
There's some infectious pathogens are known to have a trigger. Yeah, correct.
There is no cure as such, so it must.
Yes. Yes.
Can you elaborate on next generation sequencing, which is an advanced technique which is going to come on the market in the recent years?
So next generation sequencing is a technique which is next generation, right? So you can sequence at the same time several genes that you are interested in. So suppose you're doing a cancer screening, you can screen for breast cancer, colorectal cancer, cervical cancer, all those cancers at a single run. Okay, it's a higher end sequencer, but the principle essentially remains the same as previous. Yes.
Good afternoon, ma'am. As you mentioned that you have produced several types of insulin, like slow insulin, active insulin, and so on. So, what are the exact modifications in each of these types?
Okay, but so they are basically amino acid mutations. Okay, so the insulin has 2 chains, each chain has a chain, correct? Sometimes in the first acting insulin there is an as part which is replaced, which replaces another amino acid. So they are just single point mutations to make it either slow acting or addition of a certain amino acid like a glycine to make it a long acting. Also, what you can do is to this insulin, if you add a fatty acid, then its clearance from the body takes longer time. Right? So that also makes it slower. Right? Those kind of modifications.
Epigenetics, it is said that thought process can cause mutation or thought process can reverse the mutation. Is it possible?
I don't think so. Thought process, we are talking science. I don't think that will be very possible.
Is there any influence of thought process on genes?
It's more environmental factors. What you eat, your lifestyle, air. I think it's more related to that. I'm not sure what this point was.
Just to add to that, I think it's just the positivity that probably affects lifestyle.
Correct. Correct.
That's probably
Exactly.
sort of effect. I have one question for you. With this fetal DNA testing versus amniocentesis, what is the accuracy difference that you see?
Yeah. So we've done a small study for about 100 patients. So we look amniotic fluid also, mother's blood also. And we found almost statistically significant correlation of almost 95%, with 95% confidence interval, we can say that they're highly correlated. So amniocentesis problem is that in the process of amniocentesis, there is a risk of losing.
Okay. So was it like you already predicted that in today's time the diabetic disease is going to be so much and you started to study
That is what he
in
means.
2000 about the insulin?
See, I also was reading the same textbooks like you all, correct? आप लोगों ने पढ़ा है ना आपके टेक्स्ट में इंसुलिन रिकॉम्बिनेंट इंसुलिन के बारे में मतलब स्टडी ना
Yes.
पर हमारे टाइम में कभी ऐसा ऑप्शन ही नहीं था वी हैड टू बाय दिस सो दैट्स व्हाई वी सेड चलो लेट्स ट्राई इंसुलिन क्लोन करके देखते हैं मुझे जीन बनाने आता था मुझे क्लोन करना आता था तो ऐसा लेट मी क्लोन एंड सी क्लोन किया शेक क्लास पे एक्सप्रेस हो गया शेक क्लास पे एक्सप्रेस हो गया आगे क्या करूं अच्छा लेट्स स्केल अप लेट्स डू फर्मेंटेशन एंड सी फर्मेंटेशन पढ़ा था microbial. We started doing that. So it happened like that. Of course, now as a afterthought, if you ask me, diabetes is India is like the global capital of diabetes today. And that worked out. But having said that, it is very relevant. It's a very relevant medicine because patients suffering from diabetes, whoever is on insulin, it's a chronic thing. So you have to take it every day, 30 days a month, 365 days a year. Right? So if you ask me, it turned out in a good favor.
So I had a question. How and when did you decide to become a hospital?
Okay, so when I was doing the PhD, we got this project from a very small pathology lab, one pathologist approached कि ऐसा कुछ अलग टेस्ट करो बायोटेक्नोलॉजी लेके कुछ वायरस कैन बी डिटेक्ट वायरस one doctor from Apollo Hospital in Hyderabad, he reached out and he said, just like cytomegalovirus, you heard of this name? It's a virus which is present हमारे सब में हो गए फाइटो पीसीआर लोगों के सब में सीएम में होगा but in some cases where the immune system is a little compromised it can create a havoc. Now this doctor was saying that मेरे पास patient है who has cytomegalovirus but I want to know how much cytomegalovirus viral load. So that time there was no real time PCR. So that was a PhD problem, right? So I did my first experiments to count the number of viral particles. And then he said, let's find out before treatment and after treatment if there is a difference in the viral load. So small projects like this kept coming, they kept going at me and it made me think that all क्या कर सकती हूँ this medical biotechnology to make my biotechnology a relevant tool for the doctors.
Questions are there.
That's all I have.
Thank you very much, ma'am. It was truly an inspiring lecture. We could see from the questions also that students are inspired through your journey. So thank you for your profound words. I'm sure it gave a new perspective to look at the diagnostics, molecular diagnostics, genomics, and overall the power of science to improve our healthcare. At this juncture, we are going to have one performance by a team from our department. This is one team. I request volunteers, till I introduce the team, I request volunteers to arrange the table. We are going to have a performance of a street play. As ma'am explained, Jeshri ma'am, that this one for program was mainly for the public awareness. And we had gone out to the community taking the street plays. There were two street plays. One was performed on day one, and we are having another one. So let us sit back and enjoy. It will just take uh, 5 minutes, and then we are nearing the end of the session for today. So I request everyone to remain seated. I invite the team who are going to present a song from street play. Please come forward, everyone.
क्या आपको? टेंशन काहे ले रहे हो, हम है ना यहाँ। चलो आते हैं अब पति की बात पर। देखा एक ख्वाब हमने 2020 में, डीएनए को जानने सब जुटे हुए, फिर सोचा खुले, देखा एक ख्वाब हमने 2020 में।
साल 2024 जीनोम इंडिया प्रोजेक्ट भारत की एक अनोखी सोच का पड़ाव पूरा हुआ। यही है वो साल जब वैज्ञानिकों का प्रयास सफल हुआ और डीएनए जो अनजान था अब जाना जाना सा लगने लगा और इसकी हर एक बारीक हरकतों पे नजर रखी जा रही है।
तुम ही हो गौर से तुम्हें जानूं, आखिर क्यों पल में यूं असमय हो गया। क्रोमोसोम मैंने देखा, डीएनए मैंने जाना, जो राज था वो खुल गया। जीन की खुशबू जलन लग जादू, तो के बेकाबू चेंज हो गया। तुमने मेरी हाइट, तुमने मेरा कलर, तुमने मेरी आंखें, तुमने मेरा जेंडर सबसे अलग बना दिया।
अब हम जान गए हैं कि इस सब की वजह ये डीएनए ही है और इसी डीएनए को खोजने के लिए 1990 में पश्चिम देशों ने ह्यूमन जीनोम प्रोजेक्ट चालू किया और उसी के लक्ष्य कदम पे चलते हमारे भारत देश ने भी जीनोम इंडिया प्रोजेक्ट शुरू किया। अब तक 10,000 लोगों के डीएनए जांच किए गए हैं और हमारा लक्ष्य 10,000 से 100 करोड़ तक का है। ये कामयाबी हमारी सफलता की पहली सीढ़ी मानी जा सकती है।
सोचा कहाँ था ये जो हो गया, माना कहाँ था ये तो हो गया। ढूंढ रहे हैं लाजब हर रोग पे एक अनोखा किस्सा इतिहास के पन्नों में। छोटा सा पाँव है या छोटा हाथ है, अब तो समझ आ गई जिसकी ये चाल है। छोटा सा पाँव है या छोटा हाथ है, अब तो समझ आ गई जिसकी ये चाल है।
इस प्रोजेक्ट के माध्यम से डीएनए में होने वाले बदलाव और इन बदलाव से होने वाली जेनेटिक बीमारियां जैसे कि सिकल सेल एनीमिया, डाउन सिंड्रोम, एल्बिनिज्म जैसी बीमारियों पे जांच की जा रही है और इन्हीं के लिए दवाइयां भी बनाई जा रही है। हम सबके बेहतर जीवन शैली का निर्माण इसी प्रकार से हो रहा है।
तुमको जाना है तो जैसे कोई हो, जानना चाहूं कि तुमसे क्या जानूं। डीएनए में हुई वेरिएशंस की स्टडी, अब जेनेटिक रोगों का राज मैं जान सकूं। मैं अगर कहूं सपना ये हंसी, हर बीमारी पे पर्सनलाइज्ड मेडिसिन, हकीकत ये भी तो सच है कुछ भी नहीं। तुमको जाना है तो जैसे कोई हो, शौक में लगे रुके रुके, चेहरे पे खुशी रही है, बीमारियों की खाली खाली उलझन शान से हटने लगी है। लहराता परचम है जैसे बादल, प्लास्टिक में लगे जैसे रोशनी, उम्मीद की रोशनी। मैं अगर कहूँ सपना ये हंसी, हर बीमारी पे पर्सनलाइज्ड मेडिसिन, हकीकत ये भी तो सच है कुछ भी नहीं।
इस बात पर हम पूरा करते हैं हमारे सपने, लेकिन जान लीजिए ये तो बस एक झलक है, आगे कामयाबी हमारा इंतजार कर रही है। कोई बात नहीं अगर आप इस बारे में अब तक नहीं जानते, इसके आगे से इसे जानना और इसे सभी तक पहुंचाना ये भी हमारे विकास का एक हिस्सा बन सकती है। हम सबकी आशय ऐसी ही बनी रहे।
जब परेशान करे बीमारी रात हो इलाज कहीं हो आसपास आशाएं दवाइयाँ जैसे तीर है करे मुश्किल रोग आर पार आशाएं गलतफहमी अतीत सच्चाई को तू जाने दे आशाएं जगी उम्मीदें जगी अनजाना नहीं कुछ भी नहीं कुछ भी आशाएं जगी उम्मीदें जगी अनजाना नहीं कुछ भी नहीं कुछ भी
Thank you. So I call upon another team who would also be presenting a street play. I would like to announce that the first street play was presented by the students of T5 Biotech and T5 Microbiology. Now the next team, the group from SY Microbiology, all girls team.
हीमोग्लोबिन की असमानता के कारण उनका आकार अर्धचंद्रमा जैसा हो जाता है, इसलिए ये लोग क्या होता है?
ये कोई सीरियस बीमारी है क्या डॉक्टर?
डॉक्टर ये लीजिए।
अरे सॉरी,
ये लीजिए। अरे एक मिनट, क्या ये बीमारी आपकी बेटी को हुई है?
हाँ ये बीमारी मेरी बेटी को हुई है, कुछ खाती पीती नहीं है ना इस वजह।
नहीं, आप गलत समझ रहे हो। ये बीमारी की वजह ये है ही नहीं।
आपको कैसे पता है बीमारी की वजह यही है? आप हो कौन?
मैं एक साइंटिस्ट हूँ और मैं इस बीमारी पर रिसर्च कर रही हूँ। चलिए मैं आपको अपने मैजिक लैब की सैर करवाती हूँ। मैजिक लैब, ये क्या होता है? आइए देखते हैं। तो देखिए, ये है मेरा मैजिक लैब। ये देखो, आज बॉस ने एक नया आदमी को लेकर आया है। दादा देखिए आप। हाँ क्या जानते हो मैं कौन हूँ? फिर चालू हो गया तेरा। अब इनकी बात मत सुनिए।
तुम तो बड़े अजीब दिख रहे हो, तुम कौन हो?
मैं बताता हूँ मैं कौन हूँ। ये आपकी बेटी है ना?
हाँ।
आपकी नाक और आपकी नाक बिल्कुल सेम है। पता है क्यों? मेरी वजह से। क्योंकि मैं हूँ क्रोमोसोम, यानी कि गुणसूत्र। और ये जो नाक सेम है ना, वो मेरी वजह से है। यानी क्रोमोसोम ज्यादा ही पटे ही मार है। तू जो यहाँ खड़ा है ना, वो मेरे और मेरे चार में स्पेयर की वजह से खड़ा है। और तू जो ये बोल रहा है ना नाक सेम है, वो तेरे नहीं मेरे वजह से है। डीएनए की अभी कोई बोल सकता है क्या? डीएनए भाई। हाँ, मैं बोल सकता हूँ। जीन नहीं तो कौन है? हाँ, जीन नहीं तो कौन है? कौन इधर? जीन नहीं तो कौन? ये जो तुम लोग बोल रहे हो ना कि नाक से मैं तुम्हारी वजह से, वो मेरी वजह से है। मैं ट्रांसफर हुआ तभी उन दोनों की नाक से है। बड़े आए बोलने वाले, शांत रहो अभी तुम लोग। तो मैं बोलूंगी वो तुमको करनी पड़ेगी, जैसे कि हमारी बात चल रही थी। अभी तुम लोग शांत रहो, ठीक है? आया बड़ा डीएनए, आया बड़ा क्रोमोसोम। चलो भागो तुम लोग, चलो सिम कर जाते हैं।
अरे कहना क्या चाहती हो?
रुकिए, मैं आपको विस्तार से समझाती हूँ। तो देखिए, ये है डीएनए और इसका एक सेगमेंट है जीन। जब हम इसे थोड़ा और कंडेंस करें तो ये बन जाता है क्रोमोसोम। देखो, क्या वो पेड़ है? चादर ओढ़े या खड़ा कोई? कौन है जिसने मेरी ये नींद खराब कर दी? इतने अच्छे से सोया था मैं।
अरे जैसे मेरे और मेरे पापा की नाक से मैं, वैसे आप उसकी तरह ही दिख रहे हो, लेकिन आप थोड़े से कमजोर क्यों हो?
Excuse me, अब मेरा नाम इससे छोड़ रहा? मैं कहा हेल्दी क्रोमोसोम और ये तो रोगी है रोगी। मैं कहा और ये कहा बॉस क्रोमोसोम is abusing me. He's telling a lie. चुपचाप जाके बैठ जा वरना मार खाएगा।
क्या आप पहले से ऐसे हो?
नहीं, भोला भाला था, सीधा साधा था, मैं तो नादान था। दुनियादारी से होशियारी से मैं तो अनजान था। लगी चोट ऐसे मेरे भोले दिल पे, बिखर सा गया टूट के। मैंने इनके जैसा ही हेल्दी क्रोमोसोम था और मेरे डीएनए के बेस पेयर में कुछ चेंजेस की वजह से मैं ऐसा हो गया हूँ। आपकी बेटी को जो सिकल सेल एनीमिया हुआ है, वो एक जेनेटिक डिसऑर्डर है और उसके लिए भी यही रिस्पांसिबल है। मारो इसे मारो।
जेनेटिक डिसऑर्डर क्या होता है?
जेनेटिक डिसऑर्डर एक ऐसा डिसऑर्डर होता है जो जेनरेशन टू जेनरेशन ट्रांसफर होता है।
पापा, हमारे घर में जो न्यू बेबी आने वाला है, क्या उसको भी ये डिसऑर्डर होगा?
हाँ, हो सकता है।
फिर तो ऐसी कई बीमारियां होती होंगी जो जनरेशन टू जनरेशन ट्रांसफर होती होंगी, और हमें कैसे पता चलेगा?
क्यों नहीं पता चलेगा? ऐसे बहुत सारे साइंटिस्ट हैं जिन्होंने मिलकर एक प्रोजेक्ट पर काम किया है और उस प्रोजेक्ट का नाम है ह्यूमन जीनोम प्रोजेक्ट। ह्यूमन जीनोम प्रोजेक्ट ये क्या होता है? आइए देखते हैं। इस प्रोजेक्ट में हम डीएनए के सीक्वेंस को रीड कर सकते हैं और इससे हम डिजीज कॉजिंग जीन को भी आइडेंटिफाई कर सकते हैं।
Human Genome Project. ये प्रोजेक्ट एक वर्ल्ड वाइड प्रोजेक्ट है। ये प्रोजेक्ट 1990 में हुआ। इस प्रोजेक्ट के अंदर बहुत सारे देशों का कोलैबोरेशन।
ये प्रोजेक्ट बहुत सारे ह्यूमन लोकेशंस आइडेंटिफाई किया गया है। हमारा जो ह्यूमन जीनोम है वो बहुत ज्यादा कॉम्प्लेक्स है। 3 बिलियन बेस पेयर से बना हुआ है।
मैं खुद भी छोटा पैकेट बड़ा लगाऊंगा। क्या आपको पता है आपके शरीर में मेरे जैसे 3 बिलियन बेस पेयर है? 3 बिलियन?
अगर ये मशीन इन 3 बिलियन बेस पेयर को अकेले रीड करने जाएगी तो उसकी तो पूरी जिंदगी निकल जाएगी। इसलिए दुनिया भर के साइंटिस्ट ने मिलकर इस प्रॉब्लम का सलूशन निकाला है।
अभी जिनको पढ़ तो लिया पर इसका क्या फायदा? क्या फायदा? इस प्रोजेक्ट के थ्रू जीनोम सीक्वेंसिंग की हेल्प से हम बहुत सारे जेनेटिक डिसऑर्डर को आइडेंटिफाई कर सकते हैं, जैसे कि सिकल सेल एनीमिया एंड एपिलेप्सी यानी कि मेरी और पार्किंसन, मतलब हमारे ब्रेन से बॉडी का कंप्लीटली कनेक्शन हट जाना। आपको समझा क्या? आपको समझा क्या? नहीं ना, मुझे भी नहीं समझा। अरे भाई आखिर क्या कहना क्या चाहते हो? ये जीनोम सीक्वेंसिंग आखिर है क्या? बताओ बताओ बताओ। रुको जरा, सबर करो, मैं बताती हूँ जीनोम सीक्वेंसिंग मतलब क्या है। जीनोम सीक्वेंसिंग यानी कि किसी भी ऑर्गेनिज्म के कंप्लीट डीएनए को रीड कर लेना, यही तो होता है जीनोम सीक्वेंसिंग। वाओ, ये तो कितना इंटरेस्टिंग है।
बाहर के साइंटिस्ट ने काम किया है और हमारे इंडिया में सब फैसिलिटीज कहाँ?
अरे ऐसी बात नहीं है, हमारे इंडिया में कई साइंटिस्ट हैं जिन्होंने इंडियन जीनोम प्रोजेक्ट की हेल्प से 10,000 डीएनए सीक्वेंसिंग करने का गोल पूरा किया है और इसमें डिफरेंट एथनिसिटी के लोगों के डीएनए की सीक्वेंसिंग हुई है।
आप ही सोचिए, अगर हमें बीमारी के बारे में पहले ही पता चल जाएगा तो हम इसे डिटेक्ट करके मेडिकेशन के थ्रू ट्रीट कर सकते हैं। हालांकि यही नहीं, जो बीमारियां पहले ठीक नहीं हो सकती थी जैसे कि कैंसर, डायबिटीज एंड कार्डियोवास्कुलर डिजीजेस, इनका भी हल निकल चुका है और ये सारी चीजें पॉसिबल हुए हमारे इंडियन ह्यूमन जीनोम प्रोजेक्ट की वजह से। इसलिए कहते हैं अगर स्वस्थ रहेगा इंडिया
तभी तो आगे बढ़ेगा इंडिया।
Thank you, Team Info Micro. As we bid farewell to this enriching celebration, let's carry forward the flame of curiosity and passion of knowledge. I now request Dr. Kalpita Mulhan to express the gratitude. Invite her for proposing formal proposals.
It's a great hope that you have enjoyed all those 3 days. I am here again in front of you, but now this time to actually propose the formal vote of thanks. It's been really an honor and the privilege to propose and deliver the formal vote of thanks in this elite gathering of SVATV, celebrating the success of very ambitious Genome India Project. I express my sincere gratitude to today's eminent speaker, the scientist and a true Indian at heart, Dr. Archana Krishnan ma'am, for being there, gracing the occasion today and delivering the valedictory address that provided the valuable insights in the future diagnostic and really motivated the young minds to pursue the field with much more serious, actual serious enthusiasm. Thank you so much. Your talk must have led to actual realization to all those sitting in this audience how the syllabus that is getting taught in the small, modest classrooms indeed is going to have a lot of significance when you go out and actually implement it practically. Thank you so much once again, ma'am. The words are not enough to thank Dr. Mahesh Bedekar, sir, Honorable Member PPM, who suggested to have the academic celebration for the success of Genome India. Thank you very much, sir. It's this idea that stimulated the creative minds, initiated the exchange of ideas, gathered all the constructive hands together, and brought the actual interdisciplinary forum in reality with the young engineers working closely with young biotechnologists and microbiologists. So big round of applause for all the participants who participated in model making, poster making, and so on. Thank you once again for all the trust in us and the unconditional support and the guidance that Dr. Mahesh Pedekar has provided during the organization of this event. I thank Vidya Prasarak Mandal for constant encouragement and the infrastructural support that they always make available for us. The journey from just the ideation till bringing it in actual reality was such a wonderful, enriching experience for me personally as well as for all my dear colleagues at the department. I wish to thank our dynamic leader, Dr. Vilda Mansankar, Principal, B.N. Bhandodkar College of Science, Dr. D.K. Nayak, Sir, Principal, BPM College of Engineering, for the valuable suggestions and the support. Thanks are due to Dr. Suchitra Naik ma'am, Dr. Nitin Joshi sir, and Dr. Srividya ma'am for their valuable suggestions and encouragement. Let me put on record our heartfelt thanks to Up and Up India Private Limited and Rotary Club of Thane Suburban for the generous financial support for organizing this event. Thanks to Ms. Mamta and the other colleagues from the office who helped us with office communications and administrative support. A big thank you to Dr. Mahesh Patil sir, Dr. Vasi sir for all the guidance and the support for the organization of this event and publicizing this event. Thanks are due to our technical support staff, also Mr. Sagar Joshi and his team for the pandal arrangement and even our catering service providers. The special mention and a big thank you to Ms. Saili Dattardar, Ms. Urvi Shah and Dr. Ashwini Tiak, my dear colleagues from the department and all my other junior staff members who work relentlessly for making this event possible. Thank you very much. The model exhibits that got appreciated would not have been completed without the contribution from the guiding teachers from the engineering college. I wish to thank Dr. Vaishali Joshi ma'am and Mrs. Suhasini Shukla ma'am for guiding and motivating the engineering students to work on such a different idea. Thank you so much. Thanks are due to librarian Kadambari ma'am and her staff for the dockets, the non-teaching staff of the Department of Biotechnology and Microbiology, Mrs. Mohini, Sunil Dada, Vaishali Mamshi and Prachitai for working and helping teachers and their students by fulfilling all their requirements. A big thank you to all of you. The journey was made memorable and exciting all because of the enthusiastic, energetic participant students who come forward and volunteered unconditionally for making this event a huge success. Let me thank all of you all. A huge round of applause for each one of you. Thank you so much. Last but not the least, today's wonderful audience. Thank you all of you for gracing this occasion and making it a huge success. Thank you all. May I request all the participants participants from the engineering college, people who participated in street plays, people who are involved in model making and poster making, to quickly be on stage for a one quick snap with the dignitaries. And then we end this function. I request all the participants to, after this, after Pasatan, all the participants should go to Pandal. Ma'am would be visiting the exhibits. So I request all of you to quickly assemble in Pandal after this. May I request Dr. Ashwini Tirak ma'am for Pasaida.
Before everyone, I request please settle down, you can sit down. Feedback QR code will be displayed before Pasada. I request everyone to give feedback. Yes. All of you please scan this QR to fill the feedback. That is how we know your attendance.
The feedback is personal and not.
It is your feedback, not your friends. आता विश्वात्मके देवे येणे वाग्यज्ञे तोषावे तोषोनिमजदावे वसाय दानही जे खळांची व्यंकटी सांडो तया सत्कर्मी रती वाढो भूता परस्परे जडो मैत्र जीवाचे दुरितांचे तिमिरजा विश्व स्वधर्म सूर्ये पाहो जो जे वांछील तो ते राहो प्राणी जात वर्षत सकळी ईश्वर निष्ठांची मांदियाळी अनवरत भूमंडळी भेट तू भूताना चला कल्पतरूंचे आरव चेतना चिंतामणीचे गाव बोलती जे अर्णव पीयूषाचे चंद्रमेचे अलांछन मार्तंड जे तापहीर ते सर्वावही सदा सज्जन सोय रे भूमी किंबहुना सर्व सुखी पूर्ण होनी लोकी भजी जो आदि पुरुषी अखंडित आणि ग्रंथोपजीविये विशेषी लोकीये दृष्टादृष्ट विजये हो आवे जीवी एथ म्हणे श्री विश्वेश्वरा येणे बरे ज्ञान देव सुखिया जा Thank you everyone. I hereby announce that the program is over. Thank you.